instead of preventing mitochondrial disease, you will now hearing the elimination of mitochondrial disease in embryos. And it's my real pleasure to introduce Professor Juan Carlos Izpisua Belmonte from the Salk Institute.
Thank you very much, Norbert, and all the other organizers. It's a pleasure to be here. My first slide is there is no relevant financial relationships to disclose. And I'm going to tell you about a couple of methods that we're working to try to fix mitochondrial DNA, try to fix nuclear DNA, and since Norbert gave me a little bit more of time, how to try to fix cells that don't work properly. So the first thing is the mitochondria. Hon Ma has explained it very well, the problem, the background, and solutions for that with this wonderful technology that they developed a few years ago. What I would like is to remind you of how this phenomenon of homoplasmy and heteroplasmy has a role in the phenotype of the disease. So you all know that below a certain threshold, there will be no phenotype. If there is a specific number of mitochondria affected that are below this threshold, the patient will not really have that dramatic phenotype that you usually see in mitochondrial diseases. And this is the concept that led us to think of how could we just reduce the number of mitochondria. This is already published, so I will go rather quick. So by using restriction enzymes or TALEN to try to induce this heteroplasmic shift. So the model we first used was in this mouse that it has just different base that allows for a restriction enzyme to be used, in this case, APAL1. And the experiment is very simple. You just make an RNA injection with this restriction endonuclease. First, it was done in adult cells. You can see here a quite large reduction for this part coming from this mouse. Then you can do this in oocytes. You see here a reduction that is quantified here. Or you can do that, the same thing in embryos. And an animal comes out where this shift has taken place, an animal that seems to be healthy, that the subsequent generations, we are now in the 5th generation, they seem to have this reduction here, and they happen to be healthy. So that technology is quite efficient, but you have to realize that if you look at all the mitochondrial mutations that are out there, you only have 2 mutations that generate a unique restriction site for a particular endonuclease. So the majority of the mitochondrial mutations, they don't have these restriction sites. So that's why we then move to the use of TALEN that could do the same thing as the restriction endonucleases. These are some of the diseases we are focusing and developing the TALEN methodology to do the same experiment. First, as a proof of concept, it was done in a fusion experiment using mouse oocytes with human cells. And as you can see in these 2 diseases and this pair of TALENs, you can see a decrease in the mutant one that's quantified here. Now, but once we knew that this experimental setup with mouse oocytes and human cell fusion seems to be working is when we started to, let's move now to a patient. And for that, we started a collaboration with Norbert as well as other hospitals. And this is the first patient we are studying. It has a mutation, specific mutation in this mitochondrial disease. You can see here in the patient oocytes the presence of that band that indicate the mutation. And we have started to inject the talents in the human zygotes. As you can see here, if the expression of mitotalent in the human zygote is very evident and is normal, and that treatment, that injection, doesn't seem to do anything bad to the embryos. The embryos look normal, they look healthy, and you can see in this graphic here that you can eliminate human mitochondrial DNA by using a specific mitotalent. So our current and future plans are basically to try to see if this is safe to do it in human and if it is functional. And for that, our main goal is to focus exclusively right now in the lab, to derive ES from these embryos, to look at the trophoectoderm, and to do as many tests as possible to ascertain how safe this technology is and how functional the cells that derive from this embryo, at least in the Petri dish and using animals as surrogate, to see how these cells can differentiate. So the basic idea with this methodology, this heteroplasmic shift, is to reduce the number of mitochondrial mutated DNA. And as you have guessed from my talk, it's rather simple. It's just designing a couple of talents, a simple injection, and no donor oocyte is required. Now, Norbert asked Hong Ma about some issues that have been recently published and how these somehow may affect the technology that Hong has beautifully described. I think there is nothing perfect. You always have to take the best of every technology, and I would like to propose that perhaps some of the concerns that Norbert has put on the table could be better addressed by mixing both technologies. One could first do the mitotalent and after that do the pronuclear transfer. I am convinced that the mixing of these two technologies will make a safer and perhaps more efficient methodology at the end. And this is something that we are just thinking and getting it to work together with Honma and Shukrat Mitalipov. Now, that's regarding fixing mutations in mitochondrial DNA. Another aspect that I would like to touch briefly is the nuclear DNA. So we have seen during this year, even last year, that how the CRISPR-Cas9 technology has been used to edit the nucleus of a human embryo. And the message that we can extract from this first observation is that the efficiency is very low. There's a high degree of mosaicism and off-target mutations. So I think besides the ethical aspect of this experiment, we all will agree that we need better genome editing tools, more efficient, more robust technology. So just a brief pause to summarize what we know so far. So in the last couple of years, there have been different nucleases that can cut specifically in the human DNA. but they don't work alone. They need to use the endogenous repair mechanism of the cell. And as you know, there are two major pathways there. Is this one here, where you can, by using this pathway, the homology-directed repair pathway, and for many years we have been able to do knocking, gene correction, generation of reporter lines, which have been really very useful for basic biology. And there is another pathway, the non-homologous end joining pathway, which is much more efficient. It's also error-prone, but is much more efficient, and that has been used for gene knockout. They work at different stages of the cell cycle. This one here only works in cells that divide, while this one here works in cells that divide and do not divide. So mainly these two pathways have been used to modify, to target dividing cells. While these could certainly be used to modify the embryo, the zygote is a dividing cell at that moment, there are cells that do not divide. For instance, the zygote, the oocyte, or somatic cells. Most of our somatic cells do not divide. So the possibility of introducing DNA, say, in the oocyte or in somatic cells is certainly something that we don't have there yet, but could be of very practical use. So, and for that, in the last couple of years, we have started to develop methodologies in order to try to be able to introduce pieces of DNA in non-dividing cells. So here you have this particular pathway that when it's going in the error-free mode, if you bring a piece of DNA there, it will always be repaired, cut, Cas will cut, the ligase will make the join, but sometimes you have errors that introduce mutation, and when that happens, Cas9 cannot cut again. So the idea of this technology was, excuse me, let me see if this video, and it starts somewhere here. We'll just go in a moment into the next. So the idea of this technology, homology-independent target integration, is that we have similar sequences in the donor and in the endogenous DNA, and that will allow cut at the same time Cas9 to cut. And we'll introduce your piece of DNA there, most of the time in the right orientation, in the desired direction that you want, and then Cas9 cannot cut again, and you have a permanent integration of your piece of DNA. In a few occasions, it will be introduced the other way around, but again, as you can see in the video, if it moves, that we can modify this undesired direction just by designing very well the end pieces, and the donor can revert its position and be integrated in the right orientation. So this works both in dividing cells and in non-dividing cells. In dividing cells, and we use here the M-cherry or the GFP to compare with the HDR pathway. You can see that in dividing cells we have a much higher efficiency. And at least for editing the zygote, the efficiency of editing the zygote, which is a dividing cell, at this moment, as I mentioned previously, is very low. So having a technology that the efficiency is much higher certainly will be useful. But before going into this, let me just tell you one of the other applications of this technology will be in cells that do not divide. And for that, there are specific organs and tissues that you know that do not divide. For instance, the brain. When we take brain cells and we put them in a petri dish and we introduce all the machinery I have just described, you can see that the GFP is now expressed in neurons that normally do not divide. The efficiency of that is quite high. There is none in the HDR pathway, and the efficiency, depending on the experiment, can go higher until 60%. So this can be not only done in vitro, but in vivo. There are some of the tissues that we are focusing our attention: the heart, the brain, the muscle. In green, you can see how the DNA gets integrated, and as a proof of principle, we have applied this technology to a vision problem, the retinitis pigmentosa. These patients start to lose their vision early on in their life until they lose it totally. And after applying this technology in vivo, we're not taking the cells out of the eye, you can see that the mutation, in this case, this particular gene, MERKT, after being treated, the protein is produced again, and different tests indicate that normally the mutant eye will not respond to visual stimulus, and after the correction it can be responded, and these rats improve their vision. So the summary of this technology is that it's up to date the most efficient targeted knock-in method for in vivo dividing cells. And as well, it can be used for non-dividing post-mitotic cells. For this audience, perhaps it will be more interested if we can use this technology to modify the gametes or the oocyte. We have very recently started to work with oocytes and the zygote. I don't have any result to show you on the oocyte, but we have done a couple of experiments in the zygote. And as you can see by this number, these 3 experiments that we have done, and these just account for the efficiency. If we make the summary of these 3 experiments, the efficiency compared to the efficiency that we have today using the HDR pathway is really high, until 44%, and that's just the first experiment that we have started to do. So the summary of this part of the talk is that in the case of the mitochondria, we don't have a gene repair technology. I have introduced the use of restriction enzymes, and more importantly, the use of mitotalin to try to solve this problem. And in the case of the nucleus, we do have technologies, but they are very low efficiency to modify the zygote. And this technology that we call HITI could certainly be a possible technology to modify both the gametes as well as the zygote. Now, in addition to, and if I may so, Norbert, how much time do I have?
We have another 5.
Okay. So if, in addition to fixing the mitochondria and the nuclear DNA, I'd like to tell you a crazy experiment to fix the cell. So in the last, I would say, 20 years, labs around the world have been using ES or IPS to try to differentiate in the petri dish cells that go from neurons, hepatocyte, cardiomyocyte, even gametes in vitro. And I will say that even though this is the status of the field is something that we could not have imagined 20 years ago, so I should say it's a success. Mainly it's a success in that we have been able to get new disease modeling, human disease modeling in the petri dish. We have found a few drugs that could alleviate the disease, and in some cases even to improve the diagnosis. But we have to recognize that transplantation of the cells that have been generated in the lab is not taking place yet. And I would say that the major problem is that the Petri dish cannot provide the niche that the embryo normally provides. And that prevents the maturation of these cells, the proper maturation of these cells, or even getting the three-dimensional structure that the embryo needs will provide. So the cells produced in vitro are very different from the cells produced in vivo. And for that, and in parallel to the Petri dish, I'd like to introduce you this idea of the chimeras. Chimeras is something that we learn from the Greek and Egyptian technology, these monstrous creatures that were composed of cells from different animals. And they were chimeras, but chimeras until the last century, because in the last century, different researchers started to mix cells from different animals. First one was Janet Rosen with these two species of mice. After the first one, many others followed. And why do I want to mention the idea of the chimera? The idea is regarding this experiment that Hiro Nakahuchi did a couple of years ago, where he took a mouse blastocyst, eliminated a particular gene, in this case PDX1, which is a key gene for making a pancreas. So if you implant this blastocyst in a mouse mother, you will get an embryo with no pancreas. But at the same time, he mixed this blastocyst with cells from another animal, rat cells, and he asked, what will happen? Will I get a mouse with a rat pancreas? And the answer was yes. This experiment clearly indicates that cells can mix and an organ from one animal can be obtained in another animal. Today we have CRISPR, and this experiment can be done very quickly. And so we have started to do this with many other organs. Here you see again the same experiment, the pancreas from the rat being produced here. But you can do this with any organ, any cell type. Let me give you a couple of examples. For instance, if you eliminate this gene, NKX2.5, the embryo will arrest, the heart will not be formed. If at the same time you inject rat cells, the embryo goes on during normal development, and the heart is composed mainly of rat cells. Another example is the eye. You can eliminate the eye from a mouse, introduce rat cells, and you will have a mouse with a rat eye. So the percentage of chimerism could really go very high. For instance, this animal just before being born, you can see in red that the chimerism of the rat cell is quite high there. And we don't know after, when the animal is born, and after a couple of years, I will say this is the oldest animal we have, a chimeric animal today in the world. We don't even know where to put it. Our animal care people, they say, do we put them in a mouse cage or in a rat cage? It's really difficult to see what it is, but the chimerism, you can see it goes through all the tissues. But what I want to say here is that the importance of the niche. You know, or if you don't know, the rat doesn't have a gallbladder, and the mouse has it. So what we take is we take rat cells that will never make this structure, this organ, the gallbladder, and put it in the mouse embryo. And then what you see here is the mouse generates a gallbladder, but it's composed of rat cells, rat that in normal circumstances will never give rise to this organ. This speaks highly of the importance of the niche. Something that I guess it will be quite difficult to do it in a Petri dish. The niche can reprogram cells to do something that they will never be doing. So this is quite good to try to understand how a particular organ develops in the mouse or in the rat. But what about if we were to put human cells in other animals? This is an experiment that Ali Emati Bribanlu did a few years ago. This was 2006 6, I think. At that moment, our knowledge of pluripotency was rather low. And as you can see here, the integration of human cells in the mouse blastocyst was not that high, and there was a retardation of the embryos. But now, 10 years later, we have a better knowledge of human pluripotency. We have different cell types that could be introduced and test their ability to differentiate. Magdalena has shown this morning a wonderful tool where we could really, from now on, try to test the ability of all these cells here to integrate, to differentiate in a human in the endogenous niche. What we are trying to do is to put these cells in a mouse embryo, and as you can see here, in green, these cells integrate and they can differentiate in the 3 main layers, endoderm, mesoderm, and ectoderm. That was last year. There was not many cells. Now we are improving a little bit. You can see that the cells we are using can integrate into most of the tissues of the mouse. But the problem we have is that the niche, the host, is the one that dictates the size of the structure that gets generated. So if we put a human cell in a rat, the human cell, the human tissue, the human organ, whatever grows there, will sync with the size of the rat or the mouse. So we decided that perhaps we should use a larger animal, an animal that has a similar organ size as the human. And for that, the pig and the cow could be good ones. So we have started to do the same experiment, putting these human cells in a pig blastocyst. And I'm going just to finish with this. You can see that at mid-gestation embryo, this is the back of the pig. There are human cells that are growing in the pig embryo. They are not only alive, but they can differentiate in many different cell types. So the message is that this is wonderful. You are putting human cells and they are growing and differentiated in a pig. So you could use these cells now for transplantation. So this is rather false. I would say this is a dream. I have shown you the best slide, the best image I could show you. We have repeated this experiment around now 1,500 times. And this is not an experiment that you do easily between the mouse and the rat. This is a little bit more difficult, just to let you know that to put the pig on the operation table, we need 7 postdocs. So it's not like just taking a mouse and do the same experiment you do in the lab very easily. And there are many problems with this experiment, and I am finishing that I would like to highlight here. Imagine this is the pig freeway. These are all the pig cells moving at a particular speed into a certain direction to make a defined organ. And what we are introducing here is a human cell that doesn't know anything about the rules of this freeway. Doesn't know the speed, doesn't know where to enter, doesn't know how to communicate with the other. So most likely what we will get is an accident. So we really need to learn a lot of cell biology in the lab, when to put these cells in, at what moment, what number of cells, at what speed. Remember, a pig takes 3 months to develop, while the human takes 9 months. So that's already a major problem. So we are right now working in all of these details. I say details, but certainly there are major questions. So again, to summarize, I have told you approaches to fix the mitochondrial DNA, the nuclear DNA, and even to fix the cell. The most important slide is the people who do this. We have a very fruitful and strong collaboration with Honma and Shukrat Mitalipov regarding the editing of the mitochondria and the nucleus, as well as with the lab of Carlos Moraes, Ji Chiao, and Dr. Campistol in Barcelona. and the HITI technology with some colleagues at UCSD as well as the Salk Institute. And finally, the Chimera project, this crazy idea that I just mentioned, this is being developed with labs mainly in Spain as well as with Pablo Ross in California and Hong Kui Den in Peking University. And last but not least, my team and the people involved in the things that I told you today. Concepcion and Jun Wu are the person leading the Chimera experiment. Alex, Yun, and Pradeep are closely working with the HITI technology as well as with Honma and Shukrat Mitalipov, as well as Kei Suzuki. Thank you very much for your attention.